All right, so now we're live. Everybody say hello. The metabolism pathways and things, I think it's useful to go through them because it does take a couple of rounds at it to start it making sense. The key point, I think, in metabolism from our perspective in microbiology is that it allows us to get to a really functional definition of anaerobic respiration and fermentation, in that glycolysis and Krebs cycle are pretty much things that everybody's doing. And electron transport and generating protons, again, is another standard thing that everybody does, that all creatures do. And the pumping of protons and that proton gradient driven ATP synthesis, or the ADP rephosphorylation, is again a standard thing. What becomes a really cool difference is what's the final electron acceptor. And from my own perspective, I used to refer to the pathway in glycolysis in which we use pyruvate as the final electron acceptor during respiration in which we don't have enough oxygen. I was calling that anaerobic respiration. And I think I must have read that somewhere. I don't think I invented that. But it's like, no, that is not anaerobic respiration, technically. That is, in fact, fermentation. So we are fermenting when we're exercising really hard and not getting enough oxygen. Anaerobic respiration really is defined as using something other than oxygen in electron transport chain as the final electron acceptor. And so it's a bizarre thing that, we don't come close to doing that, but that's using stuff like nitrate or iron or sulfur, all these different kinds of molecules where you dump the electron. And one of the key points is, because oxygen is so electronegative, it has a greater drawing affinity. It can extract more energy from the same electrons, NADH and FADH2. It can pull more energy out of there. It can generate more of a proton gradient. And so oxygen is the best electron acceptor on the planet, I dare say. But organisms can use other things, just not as efficiently. And it's, again, one of the sort of philosophical points is that if you're a single cell and you're not carrying out development, if you get to a spot where you don't have sufficient ATP floating around, your ATP is in the form of ADP, you just kind of stand around for a while. You just start very slowly accumulating energy. It's like saving up for your favorite toy. You just start saving your nickels and dimes and wait, and then you do something. With complex eukaryotes, because I got to keep my heart pumping 24 hours a day, I got to be breathing 24 hours a day. There's muscle contractions that are happening all the time, and you just can't stop. And so I have a need for ATP that is constant, and maybe not at any one time greater than a bacterium, but it has got to be present all the time. And so anaerobic respiration probably isn't going to work very well for me because I don't have as much energy that I can extract. But certainly, fermentation is not how I can operate, whereas single-celled organisms, they probably can. Well, what we're going to turn our attention to now is, in terms of pathogenesis, something that's really important, and that is the control of bacterial growth. So there's a number of interesting little bits we'll be talking about here in this section. Different techniques that we use to control microbial growth, and probably stuff that you have, at least some of you, heard of. Hopefully we'll be able to put it into a context, and you go, oh, that's why that works. Washing hands and hand sanitizer, and things like that are what we're talking about. So one of the points that's important, and people, with the advent of antibacterial soaps and increasing hygiene, which is good, we actually are recognizing that you don't want to kill all bacteria. And in fact, it is not compatible with life to not have bacteria in you and on you. On you, yeah, you can probably go for a while, but I bet there's going to be some weird side effects of removing all bacteria from your surface. you kind of could do that if you closed up the 2 orifices, or the 3 orifices, 5 orifices, your eyeballs, your nose, your mouth, all your different orifices. Somehow you just tape them up with a little bit of duct tape. You could dunk yourself in sufficient hand sanitizer over and over and over again that you could remove the bacteria on your surface, but it wouldn't be pleasant. Inside, you really have to have bacteria. Your microbiome is critical for digestion of food. So, in terms of the bigger environment, bacteria are serving a really critical purpose. The guys are responsible for fixing nitrogen in the soil, a really critical thing for agriculture and for growing plants in general. Within our bodies and our large or small intestine, they are responsible for producing vitamin K, which is an essential nutrient. And if you didn't have bacteria doing that, we'd have some pretty serious problems, blood clotting and things like that. All right. Obviously, bacteria are responsible for allowing cows to break down cellulose, which I think is a cute trick that you and I only wish we could do. The cows need to have a fair amount of time so their stomachs are compartmentalized, and it's not easy to do, but they can eat grass or straw and convert that to sugar. And if you think about that for a moment, that is a pretty amazing thing, is to take a stick and chew on it and be able to actually get as much energy from that as you would by eating a bagel. So, thank you, bacteria. They are also responsible for all kinds of biomass recycling. Without bacteria and fungi, we'd be like literally stories of dead wood and dead animals standing around us. We can use them to carry out bioremediation, that is, the ability to eat oil and remove different toxins. Bacteria are our friends. Really, why do you want to bother them? And I think the general rule is, if they don't mess with me, I won't mess with them. But if they mess with me, look out. So obviously, we do have situations in which you really have to control bacteria. You can't just let them go crazy, because they are very definite disease-causing agents. Not all bacteria are. There are a lot. In fact, most bacteria are non-pathogenic, but those nasty ones, they can do you in pretty fast. We have various kinds of changes to the environment that bacteria can do, like converting different biomass into real acidic materials. That's not good. Algal blooms occur, and very often one of the things that this does is what's known as eutrophication, where you are removing the oxygen from the water. And if you ever had any fish, you got to have a little bubbler in there to keep the dissolved oxygen high. And so dissolved oxygen is a really good thing to have in the water if the water starts to have too little dissolved oxygen. And that happens because you've got a ton of organic material, and the bacteria are just going to town, eating it, growing, using up the oxygen, and kind of like running themselves off a cliff, but they just don't really care. You end up with, really nasty, stinky, smelly, putrefied water because there is no oxygen. So it's one of the things, like, you don't want to dump raw sewage into a lake because you're providing this great food source for all the bacteria, plus lots of bacteria as well. They will eat up all the oxygen while they're consuming this food and kill the lake. So one of the things you do is, before you dump your sewage into the water, you allow bacteria to chew on it and sort of have your own localized nasty water, and then you're dumping water into the Sangamon or wherever that doesn't have a whole lot of material in it that can be chewed up and be eaten up. Another point to think about in terms of getting rid of bacteria is that bacteria often are responsible for spoiling food, primarily. I mean, there's definitely some molds and fungi, but they're way slower growing. Bacterial growth on food, increasing the shelf life. is not a bad thing. So we do have reasons to control bacteria. So now, if we think about this, what you want to do is have some kind of method that you can kill the bacteria specifically that you want to kill, and either not kill the beneficial microbes, that could be a bit of a trick, or not destroy the stuff you're trying to preserve. So a great way to sterilize stuff is to incinerate it, but that's not going to do so good for milk or for various foods. But solid metal bits, yeah, just set it on fire. So a couple of different different ideas, and these are things we know just from basic hygiene. Wiping down tables with various kinds of killing agents, because a table, if it's constructed out of the correct material, can be pretty much inert. And if you notice, the tables in the micro lab are actually different than the tables in my cell and molecular lab, and that's because the micro lab tables are designed to have pretty harsh chemicals laid on top of them. Another thing you can do is kill stuff with heat or extracting all the water out of it, osmosis, or actually dumping a ton of water into them. Preventing oxygen from getting into the soil can help stop some things. Low temperature is a great way to inhibit bacterial growth. Antibiotics, kind of a last resort actually. Blocking oxygen, things like this. So we have then physical or chemical means that we're going to use to restrict bacterial growth. And what we're going to do is just go through some of these things, and some of them you go, oh sure, but others you may, I didn't know you could do that. All right, so these are some of the terminology that we use. And it's actually an interesting set of terms. Sterilization is a condition in which you have killed everything. There is no possibility of life on this material. So we sterilize things by steam treatment or the incinerator. And in some cases, sterilization does not mean that it's sanitary or even healthy. It just means there's nothing living on it. So there is material that is bacterially infected. You sterilize it, you still don't want to get it into you because of the bacteria toxins and things. Sanitation is where we are taking things and reducing the number of bacteria. So even though it may not be sterile, if it's sanitary, the number of bacteria is low enough that we don't have any harm. The ideal would then, if you're a true germaphobe, is you'd like to have something that is sanitary and then further sterilized. That's great. We use aseptic technique, and we've been working with this. And aseptic technique is the good practices that minimize the transfer of bacteria. And I'll tell you that, on the first week, we were looking at some of the plates that were contaminated. That was an example of not great aseptic technique. Bacteria media, depending, you can open the plate up for a few moments and not contaminate it. Most of the time when we're working with bacteria and bacterial cultures, the growth time is only a day or so. And so molds, fungi, take 4 or 5 days to get going. So you can do a bacterial experiment with some media that might actually have fungal and mold contamination, but the experiment's done so fast that you don't even care about it. On the other hand, if you've got a plate that you need to keep and incubate for multiple weeks, and we actually run a variety of different systems that do that, like animal tissue cultures. You might have the animal cells sitting in a flask for easily a week. We do some plant tissue culture where you take plant cells and put them onto a plate, and you have them on this plate for a month. You have to be super careful about aseptic technique with that, because now it's not just 24 hours and you go in the incubator and throw it out. You're going to let this stuff sit for weeks, and anything that gets on there will have a chance to grow. And I can tell you that maintaining that level of aseptic technique is not trivial. Disinfection, and what we're doing is kind of breaking down some of these terms that you know. We can disinfect stuff And this is using a really harsh chemical. So you generally don't want to get disinfectants on your hands. And disinfectants are things like chlorine. Disinfectants would be things like chlorine bleach. There are a whole range of disinfectants that are nasty things, and they do a great job of sterilizing and generating sanitary conditions, but you can't put them on living stuff. You certainly can't put them on your food. Then we have antiseptics, and antisepsis is using chemicals that are toxic, but not too toxic. And often the chemicals are diluted sufficiently. And so a perfect example here would be like our hand sanitizer. That concentration of alcohol, while it begins to dry your skin out a little bit, is not particularly nasty. It's stuff that you can use then externally. Internally is, even a bit more difficult. We've got things like chemotherapy, which is a generic term, but you're killing bacteria. And while you may think of chemotherapy for cancer, when we're talking about antibiotics, that's chemotherapy.
Like how sterile are gloves, you would say then? Are they not? This depends like how they're stored or whatever.
Well, let's see. Do we have any? Gloves are in the box. They probably are moderately sterile.
Like once the box is open and stuff.
Yeah, once the box is open, they're probably not all that sterile. But what you got then is bacteria that is from the air, but you pull them on, and if you don't touch anything, they're probably reasonably sterile. I mean, one of the things you do is take a gloved hand and put it into a bacteria plate. If you take just your raw hand and put it on a bacteria plate, you will definitely have, a finger-shaped set of colonies. So the gloves would do pretty good. If you want sterile gloves, they actually come in a sealed pouch. And there are procedures where doctors will actually take a pair of gloves and open them up and put them on very carefully. And those are sterile gloves. The easiest thing to do is to put on a pair of gloves and then take alcohol, swab them down real well, wipe them, and you're pretty good. The other thing is, though, there's no reason to have sterile gloves if you're going to be in an environment that's not sterile. Or, we do things to reduce load, and that's good. So, like
septic or whatever, just for preventive.
Well, yeah, I mean, it's like you could perform surgery in this room, but
you want to? No.
Was it?
But you want one.
Well, I would much prefer to perform surgery in the bathroom where it's all tile, and I could go in there and spray the snot out of it with bleach for a bit and say, okay, how's your appendix doing? Let's get you in there. And it's a hard surface. With the carpet, there's dust, not great. And so that's why surgical suites tend to be all tile and stainless steel, because you can sterilize the snot out of them, and that's important. You also want to have the temperature kind of cold. That prevents bacteria from continuing to grow. So in a surgical suite, they will do a serious clean on it at the end of the day, and they do a moderate clean of it in between surgeries. But it's designed, that you can really strip it down pretty well. Plus, when you go to the doctor's office, particularly these days, everything is one use. So, it's in these packages, they open it up. So that's the level of reducing the bacterial load. Because we've got two sterile hoods in the building in which they take air, we'll go through in a moment, and pass it over a HEPA filter. And if they've been maintained well, once you get them up and running, the inside of that cabinet is pretty darn sterile, where you can take a plate and open it up and let it sit there for a couple of hours and nothing grows on it. And you can work in those, and what you have to do is make sure that the air is blowing at you. You don't want to have your hands behind the sample and it's blowing stuff across. You always want to work like this, and using tools that you flame sterilize with alcohol. And, if I sit there and pay attention, I can actually do sterile aseptic technique that way. But, we don't do surgery that way. Partly, they will disinfect the body, this part, but there's stuff floating around. So, so we can think about chemotherapy as chemicals that are going to actually act on bacteria. And an important term that we can think of is we can actually break or subdivide different chemotherapeutics, different antibiotics, into being either bactericidal or bacteriostatic, which is an interesting kind of slicing definition. Is stuff that is bactericidal is killing bugs dead. Stuff that is bacteriostatic is making it so the bacteria can't grow in the presence of this chemical. And so antibiotics that we use, we will classify as either being bacteriostatic or bactericidal. And from my perspective, when doing molecular biology, I almost always use ampicillin, which is bactericidal. It is bacteriostatic. And I think anyway, what this means is that you can detoxify the antibiotic, and guys around it can begin growing on it that way. Anyway, that's what you're going there. So, here I said that thing, I kept it backwards. We can sort of carefully tease apart these terms, and the hope is they'll kill the bacteria, but not us. It's one of the points that a physician's assistant person I know was talking about last year. We were talking about the safety of vaccines, and her point was, we prescribe antibiotics to people all the time. Antibiotics have a significantly greater risk associated with them than vaccines. People would think nothing about taking a Z-Pack, and yet you might have an issue taking the vaccine. The Z-Pack has greater side effects and potential complications than the vaccine does. So relative risk is one that's really difficult for people to fully get their head around. And what this means then is that when we go to remove bacteria, hopefully it ain't going to hurt us, or you're not going to be standing around when it's happening. Hopefully it's not going to damage things that we need to keep intact. Samples, the tools. So there's things you can do, as we're about to see, that, this is a great sterilizing technique. Sorry, it'll blow the crap out of whatever it is you're trying to sterilize. If it costs a lot, that's a difficulty. Again, if it's complicated, dangerous, what this could mean is we can have incomplete sterilization. And this is a thing that happens sometimes in a variety of healthcare settings in which specific protocols for sterilizing are rather elaborate. Steps might be skipped or may not be completed effectively, and what you think is sterile ain't sterile. So having something that's a little bit simple, that is nice to use. All right, so let's go through some of the different techniques and things. One of the first things, if I was to ask you how you're going to sterilize something, you might say, hey, how about we heat it up? That's an obvious thing. So we have the autoclave. We have the incinerators. All right, the incinerators are burning stuff. So for tools, that works. But if they're made of plastic, that don't work. If they are delicate, you might actually have some problem with that. So incineration is raising the temperature up to, let's face it, above 450 degrees or so Fahrenheit. That's what causes carbon to start burning. Below that, it won't burn. That's still really, really hot for a lot of things. So what we can do is have a temperature that is going to effectively denature proteins, but not so high that it's going to completely damage stuff. But, if you think about this, so most of the bacteria that we're going to be thinking about that are pathogenic are these mesophiles. They're not going to do very well above probably 110 degrees, 120 degrees. You may not kill them, but you certainly will inhibit their growth. And if you hold it there for 120 degrees, you might very well kill them after 15, 20 minutes. All right, so what heat is doing, we know from a mechanistic perspective, is breaking down those weak-ass bonds that cause a protein to fold up into a functional state, and you denature the protein. And most proteins, once they've been denatured, they're not going to renature again, and they certainly won't renature in a way that they can actually do anything. And so moderate heat does a pretty good job. You can have another tool that we have is detergents, which are able to solubilize phospholipid bilayer. Without the membrane, the cell really is going to, A, lyse, and B, can't work. So soap and water and moderate temperatures works. I was actually over in Schilling and passed by Einstein Bagels, and they were offering a sous vide egg. And I don't know if people even know of sous vide cooking, but you can, I think we may have mentioned this, you can cook stuff at like 120 degrees. And that's not all that hot. I mean, you can absolutely stick your hands into that. And yet, if you hold something at 120 degrees, you are effectively killing everything that's in there. You are generating a completely sterile environment, more or less, because there's not a whole lot of bacteria that can handle 120 degrees. So you don't have to heat things up too high. Soap does a good job too. So we have then a couple of different kinds of rules and regs for heating. And as I say, we're not trying to incinerate stuff. So your temperature doesn't have to be crazy. But if you want to kill stuff, most bacteria, you either boil it or you put it into a steam pressure cooker. And let's just look at a couple of differences here. We had the autoclave up on the third floor, and this is essentially an Instapot. I don't know if you have an Instant Pot, but an Instant Pot is in fact a pressure cooker autoclave. And so a pressure cooker is the same thing. And this works on the basic chemical principle that as you increase the pressure, you also can increase the boiling point. So you can take liquids and raise them above the boiling point of water, but they won't actually boil. And that's nice because you don't have stuff boiling away. So you raise the pressure up, and then you rock them at 121 degrees centigrade for 15 minutes. Just remember that water would typically boil at 100 degrees. So you got a fair amount of pressure in here. The standard way to do this is to have a steam autoclave. I mean, there are other ways you could do it, but this is a real easy way, is you put a big bucket of water in the bottom of a pressure vessel that you can heat up. You turn the heat on, which is what ours do, we have big electric heaters, and the water begins to boil. The samples aren't yet at that temperature. The water boils, generates steam, and the steam has a way greater volume than the liquid does, and it begins to pressurize this tank like a big dog, in fact, to the point that you have to have a pressure relief valve that is kind of letting off some of that pressure. You maintain the pressure at several millibars or atmospheres above room atmospheric pressure, and that allows for the temperature to go up even higher, and you have a regulated temperature system in there as well, so you keep the temperature low enough that even at this high pressure it won't boil, but you can get way above boiling, and what that does is it kills everything. One of the things like with boiling is that there are endospores. These are bacteria that have sporulated into sort of a state in which they are going to have the ability to live sort of in stasis for hundreds of years. Spores can survive. And then under appropriate conditions, they can essentially germinate and hatch and come back again. This is one of the things that Pasteur was dealing with, is that he found if you took his Pasteur flask, that swan neck flask, with the wheat, grass, straw infusion, if you boil it for 10 minutes or so, you would dramatically reduce the bacteria growth, and basically the chicken stock wouldn't get cloudy for a couple of days. But if you waited long enough, it would begin to get cloudy, and that's because the endospores are able to sporulate. What he discovered was you could boil it, cool it, wait a couple of days, boil it again, and that would pretty much get everybody. And what you're doing is the first boiling is causing the endospores to recognize, hey, these are conditions that we can begin to sporulate in. They'll begin to essentially germinate and start populating after the temperature goes down. You hit them a second time, it's like, I got you this time. And so everybody has gone ahead and opened up. And so a double boil method after a couple of days works. On the other hand, the standard way, if you're not worried about endospores, is boil it for 10 minutes to an hour. That'll do it. So, you hear in Texas last week, after power outages, they had issued boil orders. And whether they need to or not, I mean, is partly nobody wants to get cholera. That's a bad thing to have. But the possibility that you have sewage contamination with your drinking water happens if the water pressure isn't high enough. You can have backflow from your potties and things, and toilets, and various kind of plumbing mishaps. If the plumbing is done exactly right, it doesn't happen, but still, you do worry about that. So they issue the boil order, and what you got to do now is boil water for 10 minutes or so. All right, so the boiling, that works, but it's not going to kill everything. And there are obviously things that are not going to be able to go into boiling water. The pressure cooker works. Again, it ends up changing the food. You're cooking it. There are a whole range of plastics that completely melt in the autoclave, which is quite hilarious to see what people like take a plastic tray and put it in there, or plastic tubes and things, and you come back 15 minutes later and it's just like this complete little mess. Yep.
Like, how good does like a washing machine do if you have like on warm or cold?
Well, a dishwasher running at hot with good soap is pretty darn good. It is not boiling, but it's like 150 degrees or so, because you actually have a heating element in that dishwasher.
Yeah, because I know it like steams and everything.
Yeah, but you caution, there's stuff that's top rack only, because there is a lot of heat down at the bottom. And we've got like a sanitary cycle where it actually does it brings the temperature up, probably not up to 100 degrees centigrade, but pretty darn close. And you open it up and there's steam coming out. That does a good job. One of the thoughts though is that case is sterile, but when you open it up, it's no longer sterile. But like if you're doing home canning, you have to be thoughtful about sterilizing. They always tell you to either bake your glasses at 300 degrees for 30 or 40 minutes, or put them in the hot cycle of your washer, your dishwasher, and take them out. Now your clothes washer ain't doing that. So you got a fair amount of soap in there, and you're doing rinses, but you put them in the dryer. It does pretty good, actually, I guess. Yeah, there are a lot of these things that's best not to think about it. So I can tell you that you can't put a lot of stuff in the autoclave, and there are other ways of doing it. Let's see. Oh, now another point is there is stuff that we need to not back the bacteria load, but you really can't boil it, like milk. Boiled milk is not great. First off, you don't want to boil it. If you can it and pressurize, like if you autoclave it, there's probably an amount of time that you can do it before it begins to actually separate out. The protein starts to fall out of solution. Typically what we do is pasteurize. And what this is going to do is not back the bacterial load. It's not going to kill everything. In fact, you may or may not have had this experience of a perfectly sealed bottle of milk or box of milk or whatever that you put into the fridge, and you come back 3 or 4 weeks later and crack that guy open and take a sniff of it, or make your roommate sniff it, or take a swig of it, or make your roommate take a swig of it, and it can go off really horrible. And that's because pasteurization is not sterilization. It is knocking the bacteria load way down, giving you a longer shelf life. And there are a variety of different things, like the bacteria that cause brucellosis, which is a fairly nasty disease you can get from drinking raw milk, that are killed pretty easily. But it's not going to completely sterilize this. So milk and pasteurization, something that actually is happening often with beer. You have a heating section where you heat it up at 65 degrees for 10 minutes or so. Ultra pasteurization is becoming the new thing. Most organic milk that doesn't go as fast as cheaper milk is ultra pasteurized, and what they're doing is heating it at almost boiling for just seconds, and then immediately flash cooling it, and you then have pasteurized milk, but it's not sterile. It's always interesting to think about. They sell milk in boxes now that are shelf stable, and they are either irradiating it or doing something with it. but it's a little bit different than pasteurization. Obviously, we incinerate stuff. One of the things, when you think about beer, you can get homebrew stuff that is not pasteurized, and there can be, depending on how clean the home brewer is, you can have bacteria that can make you pretty darn sick. I'm not sure if you've ever experienced this, but I've had the experience of having a non-pasteurized beer, not because I was hungover, but because there were some nasty bugs in there. It was not a pleasant experience of throwing up after having like 2 beers. Like, what the hell? So beers in the U.S. are generally pasteurized, or at least they're cleaned up. Now, in the past probably 40 years or so, 30 years, a lot of beer companies are talking about how it's cold filtered. I don't know, Budweiser or whatever, cold filtered beer. And they're saying, yes, it is clear. You're removing stuff with the filtration, so it's not like heat treating it, but you can remove a lot of bacteria. I suspect that cold filtered beer is, they're bragging about it just because it's something to say, but in part they're filtering out the yeast that they use to make the beer that are probably genetically engineered, genetically modified yeast that are patented, and they don't want them to get out. But this is a way we can begin to start knocking back the bacteria load. The other side of it works fairly well as well, in that refrigeration is a good way to preserve food. Now, refrigeration, if you have your experience in your refrigerator, you can definitely get stuff to spoil in your refrigerator. You gotta let it sit there for a while, but man, it'll definitely do it. Refrigeration is primarily slowing down the metabolism of stuff. It's not gonna kill any bacteria at all. It's going to slow them dramatically down. So what we'll often do is grow up some bacteria on a plate, put them in the fridge, and that way they're stable for a couple of weeks. So we've got the energy being reduced. That means the activation energy is less. It can also begin to sort of change up the structure of the membranes. There are a range of bacteria that can still operate in cold temperatures. Even the mesophiles can do it. Freezing is another good way. What you're doing there is, A, removing all the energy, but B, you are pulling the water out and turning it into ice. The water crystals can end up causing cellular denaturation. It can denature proteins, but also can rupture cells. It works, but you do end up having issues of dehydration and oxidation, which is why you have freezer bags where you put stuff in there, suck all the air out, throw it in, and some stuff freezes really well, some stuff doesn't. Yeah. All right. Let's see. The other physical mechanism, and we'll pick up here, but the idea of filtration, and we'll see there's actually some issues with filtration. But we have some things that are so delicate they can't be heat sterilized, and so you have to filter sterilize them. That's a pain, but it does work. All right, so on Monday, I promise exams back. On Wednesday, we're done with our microscopy. We're going to be doing metabolic tests on bacteria. Because I still can't get gram stains to work, and I don't want to talk about it. But that's all for now. I'll see you guys later.